Karen Peat tells us that her life took a profound turn during her third pregnancy, when she lay down for a nap and woke up a month later with short-term memory disability. 11 years after that, so now, 11 years later, she actually received an islet cell transplant as part of the Edmonton protocol. So one of the real kind of successes in regenerative medicine. And today, we're very happy that she can join us to candidly share her experience. So with that, welcome Karen, welcome Doug as well, and we very much look forward to hearing your talk.
Thank you. Thank you. I'm so honored to be here. So I'm going to go back in time. And I was told by my endocrinologist that I may not live to see my children graduate from high school. You see, I'd become hypoglycemic unaware. I could no longer tell when my blood sugar was dropping dangerously low. And to understand how I came to be in this situation, we need to jump back 30 years in time. I was in the second trimester of my third pregnancy, as already has been said. After a day of extreme nausea, I lay down for a nap. And as you've been told, I woke up a month later. When I began to wake up, thin, liquid-filled lines ran from my hands and feet. They hurt, so I yanked them out, their contents leaking onto the floor and seeping into the sheets. The room screamed. A rush of ghosts filled the room, reattaching every line and needle. The piercing noise subsided, and the lines were all taped back into place. I had been in a coma for nearly a month and was in the brain injury ward of a hospital. My stomach swelled with the growing child who had survived 6 hours of hypoglycemic seizures. When I first emerged from the coma, I did not remember that I was married with 2 children and expecting my third child. From preparing for my master's degree, I could now barely read, not remembering from one sentence to the next. I would continually lose the context of a passage and would have to reread it. It would take me 1 hour to read 1 page of the Reader's Digest. And then I would need a nap. After many more weeks in hospital, my son, Aaron, was finally born. Because of the coma, he was born prematurely, yet he weighed well over 10 pounds. With severe hyaline membrane disease, Aaron went straight into NICU. What a contrast to the premature babies there. Jaundiced and chubby, he was 10 pounds, 8 ounces. He looked like a miniature Buddha, and it was over a month before he came home. The coma left me with a brain injury, damage to my thirst center, and significant short-term memory loss. It was questioned whether I would even be able to live outside of an institution. Early in my recovery, I needed assistance looking after our 3 young children. Having found me in a full-blown seizure, swollen and bloated, my husband Doug ended up with post-traumatic stress disorder. It was hard on both of us, as I had forgotten most of our marriage, and both of us had to work very hard to rebuild our relationship. I was also very insulin sensitive, requiring a very, very low dose daily. Dose of insulin, of course. As the years went on, my blood sugars became more and more unstable and continued to fluctuate wildly. 4 separate times, I had to be taken from Superstore in an ambulance as I had collapsed in a writhing heap. Once again, overcome by a grand mal seizure caused by low blood sugar. My husband was instructed by our doctor to be always within 1 hour's reach. He shares that during those years when he went to sleep, he never knew whether he would wake up to find that I had simply perished in the night. This situation also had a devastating impact on our children. One morning, Dara, our eldest, 9 years old at the time, came down the stairs to find me unconscious and seizuring on the kitchen floor as liquid sizzled onto the hot element of the coffee maker. She had to be the one to call emergency. This was not the last time Dara would have to call 911. In fact, we ended up with a whole collection of small, cute white teddy bears from the first responders that they gave to children in crisis. As time went on, I became increasingly insulin sensitive. Slight variations in diet, exercise, or mood could send my blood sugar plummeting. My husband became an expert at spotting dropping blood sugars, treating them, or administering glucagon. We were going through 3 to 4 glucagon kits per month, and you know that's expensive. Living with both a memory disability and hypoglycemic unawareness, I would often find myself in trouble. One moment I'd be fine, the next I'd be mid-seizure, shaking violently and vomiting before collapsing into an incoherent mess. Security guards and police officers would assume it was a drug-related event and carefully gather my children for yet another police car ride to the hospital. My children and husband suffered lasting trauma as the seizures continued and brain injury symptoms became more pronounced. My hopes for any sort of career or operating a vehicle were painfully abandoned. So I refocused my attention on raising my children and creating a place where their friends were always welcome. In desperation, my husband and I sought the help of one specialist after another. Then one summer afternoon, we entered the cluttered, book-laden office of a white-haired, wizened endocrinologist. Slowly, he unfolded a promising experimental procedure, the Edmonton Protocol. He thought I might be eligible. Perhaps if islet cells could be transplanted from a donor into my liver, insulin could be produced there. This could decrease or completely cancel the need for additional insulin. My husband and I accepted the offer on the spot. After the appointment, we laughed and cried in awe. Perhaps our nightmare was ending. We knew this procedure would be costly, for it would involve significant time off work for Doug and arrangements for our children when we were in Edmonton. When I was first considered, we went to Edmonton for a month of dizzying tests to see if I was a viable candidate. We were relieved when I found out I was accepted. We knew once we received the phone call that a donor had been found, we would have only a few hours to get from Vancouver to Edmonton. So we began to prepare. A few days before Christmas, we received the call. I tossed some clothes into a suitcase, packed for my husband and youngest, Aaron, now 13, and a friend dropped us at the airport where we met my husband, Doug. Arriving at the hospital, I received the islet cell transplant the next morning. We knew that the follow-up would take the next few weeks, a long time to be separated from our teenage daughters back at home. Over the next weeks, kind family members kept an eye on our girls who had been left behind over Christmas. Our Christmas was spent in a hotel room. Me, Doug, and Aaron Even though we were separated from family, it was a great Christmas. The transplant was the best Christmas gift we could imagine. Receiving this medical intervention saved my life. Experiencing professional aid of the doctors and nurses is a high honor, and the staff were so compassionate. One thing did bother me. After the procedure, we saw pictures of other transplant patients eating cake and other decadent desserts. Then and there, I determined to remain on a diabetic regime and continue to take a small dose of long-acting insulin. At first, this was to keep me from being anxious, but it made sense to me to do my part and not to stress the transplanted islet cells. Initially, the anti-rejection medication played havoc with my digestive system, leaving me in severe pain. Walking my dog for long periods was my only relief. Finally, the drugs were switched and the pain disappeared. It took some time to ensure that I was on the right medication and dosage. I have been told that I am now the longest single transplant recipient. Other recipients have required 2 transplants. For many recipients, the protocol has been changed to include a maintenance dose of insulin. Because of the transplant, I could now exercise freely without fear of collapse or seizure. I could vary my diabetic diet without fear of collapsing. Dara, my eldest daughter, could relax her hypervigilance over my behavior and food consumption. Slowly, my family learned to relax after years of anxiety and fear. It has been said that hardships either break up families or bring them closer. We have a close-knit family who is endlessly grateful for my continued good health, and I was able to expand my interests. If we can improve the lives of diabetics so that the constant fear of seizures, desperate thirst, and other horrible side effects are no longer experienced, why don't we? I have taught children and re-engaged in the studies of counseling and theology. I've worked alongside women in the downtown East Side as they sought relief in their struggles. You may remember that I was told I would never see my children graduate from high school. Well, I have seen them all graduate from university, marry, and we have wonderful grandchildren. So let's ask a simple question. What is a healthy human being? Webster's defines humanity as the quality or state of being human, but I need more than that when the alarm clock rings. Embracing my humanity goes beyond mere existence to engagement with the people, environments, and events around me. I could not do that if I was constantly preoccupied with the impish capers of my blood sugar levels. So I hope sharing my story with you will serve as a small thank you for all the research that takes up your days. Through your hard work, day after day, I and thousands of others like me have a hope for a future, to survive and live a fulfilling life. So how do you measure such a gift? The Edmonton Protocol is but one example of life-changing research. Before, I was facing a very short life and resigned myself to love my children the best I could with the time I had. I never knew, my family never knew, when low blood sugars and seizures would finally claim my life. Because of research like yours, I've celebrated with pride my children's graduations, danced at their weddings, held and hugged my grandchildren, camped and hiked and experienced live symphonies, walked my husky, and savored so many other life-giving adventures. Doug and I have now been married 36 years, and despite all the challenges we faced, we're still holding hands. So when you return to your regular rhythms, I invite you to remember. Remember the impact you are having in the lives of people. Remember how your work provides hope, life, and a better future. On behalf of my family, we thank you from the bottom of our hearts.
Thank you very much, Karen, for that powerful and compelling and fortunately happily ending story of from the perspective of someone who's lived through it almost seems like 4 or 5 lives worth of struggle, and to come out on the end of it hopeful and happy and with surrounded by a wonderful family. That's a compelling vision for the future for people like us who do research into regenerative medicine. So thank you very much for that.
You're welcome.
All right. So if you please hang around and we'll have a Q&A session at the end for both you and for Corinne. And now it's a pleasure to introduce Corinne. She is Professor Corinne Hoesli is the head of the stem cell bioprocessing lab at McGill, co-director now of TCell, which is the Quebec Cell Tissue and Gene Therapy Network, and a member of the exec of our unit, the McGill Regenerative Medicine Network. By training, she's a biochemical engineer focused on bioprocess development, stem cell culture optimization, and the engineering of encapsulation devices. She's been her work has been focused on developing bioprocesses to both produce and to render viable therapeutic cell transplants to treat diabetes and to treat cardiovascular disease. She's working on how cell culture materials impact stem cell and progenitor cell differentiation and function, and helping to build them into useful therapeutic products. She has lots of collaborations with companies building cell-based products at scale. She's involved with the Canadian National Research Council to help tailor bioreactor design and to improve operational parameters to make these cells and larger constructs more viable. So she's been working to improve cell survival following transplantation. She's been building vascular biomaterials and 3D printing methods to generate vascularized tissue. In the last 5 years, she's published over 20 articles with her team in the top bioprocessing engineering journals. She holds a number of patents related to this work, and I think her leadership has clearly emerged in the area. She's been the recipient of many awards, including the Martin Sinecor Outstanding Young Investigator Award from the Engineering Conferences International and Biogen IDEC, and she was given the Étoile Effervescence Award from Montreal in Vivo. She's an amazing colleague, driven to succeed, driven to help her students succeed, driven to help her colleagues succeed, and driven to make a difference in the lives of patients. So with that, I will invite Corinne to tell us a bit about her work.
Thanks a lot, Terry. And Karen, I think the kind of talk that you just gave is what the students need to, remember why we're doing this kind of work, and myself as well. It was really amazing. So I really thank you for that. And I think we need to hear more of that over time. Like sometimes we get lost in the trees and we forget about the forest. And this is I really appreciate that. Donc, j'ai mis les titres de mes diapositives en français pour ceux qui seraient francophones, mais la présentation va être en anglais. Mais à la fin, si vous avez des questions en français, pas de problème, on peut discuter en français. All right, so you've noticed from Terry's, I forgot about how long my biography was, so next time I'll make it shorter. But basically, I'm an engineer, but I love stem cells. I love cell therapy. I love the idea of using cells as a powerful medical tool. It's one of the most powerful tools because cells in our body already accomplish all these marvelous functions. And one of those is the potential to secrete hormones like insulin, and that can be used to treat type 1 diabetes. All right. So, but I'm taking this perspective of an engineer that's trying to make this as a product that we can, produce in a bioreactor and then distribute to the clinicians that will then transplant this into the patients. And this is not a simple feat. Okay. So, as Karen just mentioned, type 1 diabetes, it is a burden on daily life. Even if you're not susceptible to these hypoglycemic episodes, it's something also that often starts young, so not always, but it used to be called juvenile diabetes as well. But basically, the idea here is that these patients and people like Karen, their beta cells in the pancreas, which are the cells that produce insulin, they have been destroyed, usually by the immune system. And an amazing life-saving therapy is to replace the insulin those cells produce. But that means that the patient has to do the job of the cell. Basically, the person has to figure out what's the blood glucose level and then inject the appropriate amount of insulin. And that is very difficult, even with insulin pumps, even with algorithms. It's very difficult to achieve that balance that the cells are absolutely expert at doing. But Canada is a leader in that field. So it was in Canada that insulin was discovered. And from there, insulin became manufactured first from animal pancreases, right? Like we would isolate the insulin from pigs or bovine pancreas. And eventually, with the discovery of how DNA works, there was a small company at the time, Genentech, that developed insulin as a product that you can make in a bioreactor. So the idea is you can take the insulin gene and trick bacteria or yeast to actually make the insulin. And then you have a bunch of insulin in your bioreactor, you purify it, and then that insulin you can then use for injection. So that was life-changing for people with diabetes. It used to be a death sentence, but or at least you only had a few years to live under a very strict diet, but insulin saved lives. And that came from a Canadian discovery. And then, as you just heard, so after that, there was a lot of more, research to figure out how the balance of glucose happens with glucagon, for example, counteracting insulin. And then in 2000, there was this publication from the Edmonton group, and several groups around the world had tried to develop transplants to treat diabetes by transplanting these insulin-producing islets that contain the beta cells. But it was in Edmonton that they figured out how to do it reproducibly with higher success rates with the proper immune suppression medication, for example, and the right doses of cells. So that's amazing, but it still is not something that everybody has access to, because when you receive these transplants, you have to be placed under lifelong immune suppression. And so as a clinician, you have to weigh the pros and cons, the side effects of the immune suppression versus the risk of that the diabetic state poses. And that's why it is usually people with brittle diabetes with hypoglycemic events, such as Karen, that are eligible for that therapy. And so as a consequence, there's only a few thousand people worldwide that have received that therapy since that publication by the Edmonton group. So the procedure that Karen described is the following. So first you have to identify a donor that is matched as close as possible to the recipient. So you need a list, you need to have people that will agree to donate their pancreas, but these are people that are brain dead. So this is not a living donation situation. So essentially, the person has to have communicated with their family that they're willing to give their pancreas or their organs, and the family has to agree to it. And so it's a difficult decision often, because these people are often still maintained on life support, but they are brain dead, and then you make the decision to disconnect that life support and to donate those organs. And then, because the pancreas, it contains not just these islets, but a bunch of other cells involved in digestion. And then you have to isolate these islets that produce insulin, but you have to do it in a way that you don't release all these other enzymes that can destroy the islets. So you have to be efficient with that. You have to do it on the spot. And then you have a certain short time frame to transplant those islets in the recipient to get the recipient in. So this is a complex process in terms of finding the donors, matching them to the recipients, doing the islet isolations in a timely fashion, and then doing the actual transplant efficiently. But the beauty of the procedure is that the transplant itself is, let's say, a minor procedure in the sense that it's only a few milliliters of cells that you need for restoring glucose control. Because in our pancreas, the pancreas is about 100 milliliters, but only a few milliliters of that are actually the pancreatic islets. So if you isolate those, you can infuse them into the liver, they lodge into the blood vessels, and there they can become revascularized, and essentially have like a pancreas in your liver. And when it succeeds, it is amazing in the sense of remaining in the proper blood glucose range, even if you compare it to an artificial pancreas, meaning an insulin and glucagon pump, where you would have like an external system that administers insulin and glucagon. Even that cannot match how well the glucose can be regulated with these islets. Like I said, they're the experts at that, and nature has evolved that expertise over centuries and millennia. Okay, so how can we make that treatment that can be life-changing accessible to more people? First, we need a cell source that likely wouldn't be sourced from a brain dead donor, because we will never meet the demand with that. And so there's possible sources like stem cells or xenogenetic islets, for example, from pig. That might be a potential source. But basically, we need a source that is well defined and that we can grow or isolate in large quantities. And then we need to make sure that when we transplant the cells, they will survive more efficiently, because even with the current transplants, not all of those islets, and actually a minority of the islets, are surviving. And then if we really want to make this accessible, we have to find a way to avoid the need for immune suppression, because, as I said, that also has consequences of abating the immune system is not something ideal to do to anyone. And then finally, even if you are able to do that, you need long-term function. So you need to deal with how the graft will perform decades later. And finally, all of that has to be done at a cost that our healthcare system can afford, especially in Canada with a universal healthcare access. So today, in the time I have, I will talk a little bit about our lab's effort, and actually efforts across Canada and in the world to source these insulin-producing beta cells from stem cells. And then in a second time, if I can get to it, I will talk about the idea of encapsulation to protect the cells from the immune rejection. Okay, so in pregnancy, the child is formed from what is called a pluripotent stem cell. So it's one cell, which is totipotent first, and then it becomes specified, and then you have these pluripotent stem cells that will give rise to all the cell types in the body. And you can imagine that the beta cell that produces insulin is one of the most specialized cells you can imagine. So it's not a simple process during pregnancy and during embryonic development to get there. And it's even harder to do the same thing in the lab, as you can imagine. But the advantage of using these pluripotent stem cells is that you can grow them to virtually unlimited amounts. And you can source them either from discarded eggs from in vitro fertilization, but nowadays you can also take a skin or blood sample, reprogram the cell into a pluripotent state, and then take those cells, grow them up, characterize them, and then differentiate them down the pathway. But like I said, this is very complicated. So in the embryo, the differentiation needs to go through many different stages, and there's morphological changes, there's other tissues evolving around it, there's cell-cell signaling. It's a very complicated process. But by studying how this process happens, especially in mice, and also a little bit in humans, but as you can imagine, this is very difficult to study in humans, companies and researchers were able to develop this process of differentiation through 7 stages. And I won't go through all the details here, but one of the critical stages is this stage 4, this pancreatic endoderm, from which all the pancreatic cells will arise. And you need the expression of a transcription factor called PDX1 for that. And for the beta cells to arise, you also need the expression of NKX 6.1. So I will just highlight those 2 as a critical marker at stage 4. And then at stage 7, eventually you actually maintain the expression of PDX1 and NKX 6.1, but you start also getting the expression of insulin and other mature beta cell markers. And it will be important at the end that these cells can respond to glucose so that they don't secrete insulin when the glucose is low. Obviously, you want this to be glucose responsive. So you can see this as, from a developmental biology perspective, but you can also look at this from a process development perspective. How do you do this in bioreactors or chemical reactors as an engineer, right? Like how do you take this very complex process and make it feasible for manufacturing? Okay, so we were very fortunate to collaborate with Tim Kiefer at UBC. He was a mentor throughout my Ph.D. as well. And I spent some months, as well as Jonathan Brassard in my lab, learning how to do this differentiation protocol. Because like I said, we're an engineering lab. We're not the ones that are going to innovate on the developmental biology side, but what we were interested in is understanding how this process works so that we can then address the key manufacturing challenges associated with it. So Jonathan has been able to replicate and even modify this protocol that the Kiefer lab had developed. And right now, the way it works is that up to stage 4, we culture the cells in planar cultures, so like on a petri dish, basically. And then after that, they become aggregated because these pancreatic islets, they're in cell clusters. So we aggregate them using these agar-wells made by Stem Cell Technologies, a company in Canada. And then we culture them in suspension, so in a stirred system. And anytime as engineers we can culture things in suspension, we're happy because it provides better mixing, better distribution of nutrients, and it allows more easy sampling and control of the system via probes. And you can see on this slide that at the end, so first of all, at the stage 4, as I mentioned, we can get the expression of PDX1 and NKX 6.1. When we're quite successful, it's around 75% of the cells that will co-express those markers. And then at stage 7, you start seeing the expression of C-peptide. So C-peptide is co-produced with insulin. Actually, insulin is a larger protein and it gets cleaved, and one of the cleavage products is the C-peptide, and the other part is the mature insulin. But we don't measure insulin because there's insulin in the culture medium. So that's why we use C-peptide as a measure instead. So you can see that the cells, they express C-peptide, and some other cells also express glucagon. Those are the alpha cells. So it's kind of the counterbalancing act that we have. And importantly, we have very little of the cells that co-express both, because those aren't really the cell type we want. We want the ones that are expressing the insulin, and also in these islet-like clusters, it's actually good to have these glucagon-producing cells as well. Okay, and then we stain the clusters for ditisone, which is a marker that it interacts with zinc, and zinc is found in mature insulin granules. So we can see that we have a pretty good product, at least close to the state of the art. And in fact, we can even put these cells into encapsulation devices, which I will talk to you later, and we can still maintain the expression of C-peptide and glucagon even in those encapsulation systems. But I'll get back to that a bit later. I see that I'm short on time, so I might go a bit fast. Okay, so there's a student in my lab that's been looking at this process and looking at the aggregates that we're forming, and there's a few things we notice. So when you put the aggregates in suspension, one of the problems that you see is that they start merging together. And when the cells merge together, what happens is that oxygen will be higher in concentration at the surface, but as the cells consume oxygen, you'll get lower oxygen in the center. And because of that, this aggregation is not ideal. So one of the consequences of that might be this drop in the cell number that we're seeing over time. And this is actually not very reported in the literature, but basically you get some cell losses from stage 4 to stage 7. So, and also another contributing factor that you might have in these cell losses is that when you put the cells in suspension, for example, in a bioreactor like this one, you will see that there are areas, because you have to mix the suspension to keep the cells in the mixture, and then you have to have a good distribution of the nutrients. But what you will see is that this impeller that's rotating, it will impart some possible damage to the cells, especially in the areas highlighted in red. That's where the fluid movement, there's the highest gradient in the velocities, which imparts shear stress. So we had several questions. So our idea was, what if we encapsulated the cells in these microbeads so that we could protect them from this shear stress effect? And as a bonus, we would avoid the cells colliding and merging and forming those larger aggregates that would have the low oxygen in the center. So then we asked ourselves different questions. If we do that, maybe there's advantages, but there can also be disadvantages, like maybe the encapsulation system would hinder the differentiation, or maybe insulin wouldn't be produced as efficiently, and maybe it might be difficult to recover the cells from those capsules. Okay, and when you encapsulate the cells, there's different ways to do it. But if we want to scale this up and produce, the 1 million islets that we need per patient, we need it to be scalable. So during my PhD, and my students now also are developing these highly scalable encapsulation methods, where actually we're making a vinaigrette. We're making an emulsion. So essentially what we're doing is we're putting the islet-like clusters together with a hydrogel, and we're making little droplets in oil. And then we just solidify the droplets during the emulsion so that we can get gel beads in the emulsion. And then we just have to separate the beads away from the oil. And if you want to know more about it, you can read these papers cited there. if you have a stirred emulsion like this one, if you're just stirring your vinaigrette, you get all sorts of different bead sizes, which has its pros and cons, but this is extremely scalable, right? You can make millions of these droplets very fast, millions of beads, and the cells survive the process. If you want to make uniform beads, you can use this microchannel device instead. So it's the same idea. You're making an emulsion, but instead of making it in a stir tank, you're passing the material of the gel with the islets through channels. And then actually in this case, they're just floating up in the oil and getting cross-linked at the same time, so you can get gel beads. And actually a little video here. So it's also pretty scalable. You can see just from one channel, you can make a lot of beads in a short period of time. And each of those beads on average might contain one of these islets. And you can get very uniform droplets this way. Okay, but what happens to cells when you put them in the beads? So do they survive? Are they happy? So Ariana and Florent demonstrated, yes, the cells are happy, they survive, and they can also be recovered from the beads with high viability, which you see down here. So the green is the live stain and the red is the dead stain, very little red that you can see. So that was good. When you look at the distribution of size, remember how I said like the aggregates of cells, they will fuse together? Well, the beads will prevent that, because if you culture the cells in the beads, they can't really fuse together. And so you don't get the increase in volume that you see with the suspension culture purely. And you still maintain the expression of C-peptide and glucagon, as well as the ditazone staining. And you can see also the difference in size. You can see how the ones in suspension, they merge together. But the ones that were encapsulated, they didn't merge like that. So we have more control over the size of these cell clusters. Okay. And then obviously we want to make sure that they can secrete insulin. So we also check that they're responding to different glucose levels. And importantly, they increase their level of insulin secretion when they're at high glucose, but then they return to lower levels of secretion at low glucose. That's quite I know it's a bit hard to see, but you can see it's going from less insulin secretion at low glucose to more insulin secretion at high glucose, and then back to lower insulin secretion at low glucose. And you can transfer that into a stimulation index. So the stimulation index isn't much different if you encapsulate the cells in these beads. So glucose responsive insulin secretion is maintained. And the last question is, can those beads actually be used in bioreactors to scale up the culture? That's the work in progress by Ariana. Okay, so in the interest of time, I have to skip, unfortunately, that part. And just briefly want to mention the idea that not only can you use encapsulation to scale up the cultures, but you can also use encapsulation to protect the cells from the immune system. So the idea is that, those beads that we formed, or this material we're putting around the islets, it can block antibodies and immune cells from accessing the encapsulated islets. And then you would have to find a way to transplant those encapsulated islets and get them to survive. I'm going to skip that. This works quite well in animals, in small animals, rodents like mice. We can cure diabetes with encapsulated islets in mice for a year. But so far, it's been very challenging to reproduce those successes in larger animals and especially primates, although some groups are getting close. And one of the reasons for that is the scale. So it's something that often in biology we don't think about, but when you scale something up, you're increasing the volume, you're increasing distances. And as engineers, we know that that changes the time for diffusion of molecules to cells, for example. So in the body, our body solves that problem by very dense vasculature around the islets. But when you encapsulate the islet, the vasculature can't get to the islet, and then the islet doesn't get the proper nutrients, especially oxygen it needs to survive. So as if you want to engineer these encapsulation systems, you're going to have to try to bring oxygen as close as possible to the islets. And you will also have to find a way that insulin can get into the blood vessels, because without that, there's no treatment, without insulin going into the bloodstream. So we're using various 3D printing methods to try to bring vasculature into these encapsulation systems. And again, because I have very little time, I don't have that much time to talk about it, but one really cool way to do that is that you can actually print vitrified sugar. You can imagine almost like caramel, where you can print like really fancy shapes, you know, out of caramel. You can see that on dessert sometimes, but you can actually do that also with a 3D printer. And then you can get fancy different, you know, kind of vascular templates. And then you put your cells around that template, and you can look at how the cells survive around the vasculature. You can model that with numerical methods. And that's it. So these are the kind of things that we're working on, and I'd like to thank my lab, because they're the ones that are actually doing the work. And you see them here, not all of them on the picture, but also many collaborators without whom this work wouldn't be possible. And I'd like to highlight Médicaments Québec, because they're the ones that have been helping us scale up this production of these stem cell derived islets. And to conclude, I'm looking for students. So if you're a student on the call, I'd be happy to get your CV. All right, thank you.